Okay, let's dive in. It's pretty amazing when you stop and think about the simple things, like hearing your favorite song just pop up on the radio, or just walking across the room without even thinking about balance feels effortless, right?
Totally effortless.
Yeah.
But behind the scenes, it's anything but simple. There's this incredibly complex, microscopic stuff going
Yep,
on.
absolutely. And that's what we're digging into today. These tiny unsung heroes behind hearing imbalance. We're talking about hair cells.
Right. And not, the hair on your head,
No, definitely not. These are super specialized cells deep inside your inner ear. They're technically called mechanoreceptors.
which is just a fancy way of saying they sense physical movement or pressure.
Exactly. Our main source for this deep dive is from excerpts of Hair Cells, Architects of Hearing and Balance.
It's a good title. Architects. Because they really are constructing our perception piece by piece. They're the crucial link, turning physical vibrations or movements into signals the brain understands. Sort of like the neuron cells of the inner ear, in a way.
Yeah, the translators. So our mission here is to unpack how these little architects actually work. We want to see the elegance the source talks about, the nuts and bolts of it.
And understand why they're so vital. I mean, how you hear anything, how you stay upright, it all comes down to these cells.
So tiny, yet powering two fundamental senses. It's wild.
And what's cool, as the source points out, is you've got hearing hair cells and balance hair cells. They live in different spots in the inner ear,
Right.
but the basic way they work, pretty much the same principle. It's very efficient biology.
Okay, so a shared foundation. Let's tackle hearing first.
Yeah.
The symphony of sound, as it were. Where do these auditory guys live?
Okay, so for hearing, you need to picture the cochlea. That's the snail-shaped structure deep in your inner ear.
The classic spiral?
Huh? And inside the cochlea, there's this specific strip of tissue called the organ of Corti. That's where the auditory hair cells hang out.
Organ of Corti. Got it. So, how does a sound wave, which is just vibrating air, become something the brain registers? Walk us through the steps from the source.
Right, so it starts outside, obviously. Sound wave hits your eardrum, makes it vibrate.
And then those vibrations get passed along by those three tiny little bones in the middle ear.
The malleus, instachus, and stapes.
Yep.
Like a tiny little drum kit passing the beat along and actually making it stronger.
Amplifying it, right.
Yeah.
So the last bone, the stapes,
It pushes on a small membrane, like a little window into the cochlea called the oval window.
and the cochlea is filled with fluid.
Exactly. So when the stapes pushes, it creates waves in that fluid, like dropping a pebble in a tiny coiled pond.
Okay. Ripples in the cochlear fluid, then what?
Those fluid waves travel through the cochlea and make another membrane inside, the basilar membrane, vibrate up and down.
Ah, okay. And the hair cells are connected to this basilar membrane.
They're sitting right on top of it. And this is where it gets really cool. These hair cells have these little bundles of stiff, bristle-like things sticking up. Those are the stereocilia,
Not actual hair, but looks like it under a microscope, I guess.
pretty much. And these stereocilia bundles are positioned just under another membrane, the tectorial membrane, which kind of sits over them.
Okay.
So as the basilar membrane bounces up and down because of the fluid waves, the hair cells ride along with it. And their little stereocilia get bent or sheared back and forth against that overlying tectorial membrane.
So the vibration causes a physical bending of these tiny hairs.
That bending is everything. It's the critical moment.
Yeah.
Because at the very tips of these stereocilia, there are these minuscule little gateways like trapdoors, ion channels.
Right on channels, so gates for charged particles.
Exactly. And tiny protein links, like little springs, connect the tip of one stereocilium to the side of the taller one next to it. When the bundle bends one way, these links get stretched.
And they pull open the trapdoors, the ion channels.
Bingo. They physically pull open these ion channels. It's direct mechanical action. Super elegant.
Wow. Okay, channels open. What happens then?
Well, the fluid surrounding the tops of these hair cells in that specific part of the cochlea is really rich in potassium ions.
Yeah.
Positively charged potassium.
So when the gates open,
Potassium ions flood into the hair cell down their concentration gradient.
adding positive charge inside the cell?
Right. Which changes the electrical state of the cell. It depolarizes it. That change is the electrical signal.
The translation from mechanical bending to electrical pulse?
That's it. This electrical signal then causes the hair cell to release chemicals, neurotransmitters at its base.
And those chemicals signal the nerve connected to it.
Exactly. They excite the auditory nerve fiber, which then fires off a signal along the nerve pathway towards the brain.
And the brain interprets that pattern of signals as sound, music, speech, noise, whatever.
All starting from that little bend It's kind of mind-blowing that your whole experience of sound hinges on that tiny mechanical event.
Truly. And the source mentioned something about pitch, right? How do we tell high notes from low notes?
Yeah, that's another clever bit. The basilar membrane isn't uniform. It's stiffer and narrower near the entrance of the cochlea and wider and more flexible towards the inner end, the apex.
So different parts vibrate differently.
Exactly. High-frequency sounds cause the stiff part near the beginning to vibrate the most. Low-frequency sounds travel further down and make the floppy end vibrate more.
So the location of the vibrating hair cells tells the brain the pitch.
Precisely. It's called tonotopy, a place map for frequencies along the cochlea. That's how you know a piccolo from a tuba.
Fascinating. The structure itself encodes information. Now, you mentioned there are different types of these auditory hair cells.
Right. Two main kinds doing slightly different but crucial jobs. Inner hair cells and outer hair cells.
Okay, inner hair cells.
They're the primary sensors. Think of them as the microphones. They do that main conversion we just talked about. Bending causes signal. And something like 95% of the fibers in the auditory nerve connect directly to these inner hair cells. They send the main message to the brain.
Okay, they're the main reporters. So what about the outer hair cells? What's their deal?
The outer hair cells are, well, they're amazing. They're like the sound engineers of the cochlea. They also get stimulated by the vibration, but they do something extra. When they get that electrical signal, they physically change their length.
Wait, they move? They get longer and shorter?
Rapidly. It's called electromotility. They literally contract and expand with the rhythm of the sound stimulation.
No way. Why? What does that do?
By pushing and pulling on the basilar membrane as they change length, they actively amplify the vibration of that membrane, especially for softer sounds.
So they boost the signal.
Yeah. They make the whole system much more sensitive, allowing the inner hair cells to pick up on really quiet sounds that they might otherwise miss. And they also help sharpen the tuning. They make the peak of the vibration on the basilar membrane more defined.
So they make us hear quiet things and help us tell similar frequencies apart better? That sounds critically important.
Oh, it absolutely is. It's like having a built-in amplifier and equalizer right there in your inner ear. Without the outer hair cells working properly, hearing is much less sensitive and much less sharp.
Wow, so it's this intricate dance. Fluid waves, bending stereocilia, ion channels, electrical signals, neurotransmitters, plus this active amplification, all just to allow you to hear the world. Incredible.
It really is a marvel of biological engineering. And like we said earlier, that core trick turning a physical push or pull into an electrical signal, it's not just for hearing.
Right, time to switch gears to balance. The vestibular system uses the same principle, but for different inputs.
Exactly. These vestibular hair cells are in a different set of structures in the inner ear, separate from the cochlea. We're talking about the three semicircular canals and the two otolith organs, the utricle and the saccule.
Still filled with fluid, still lined with hair cells.
Yep, same basic setup. Fluid-filled chambers with specialized patches of hair cells inside. But the mechanics are adapted to sense head movements and gravity, not sound vibrations.
Okay, so how do they detect, say, turning your head? Like nodding yes or shaking no?
That's the job of the three semicircular canals. They're oriented basically at right angles to each other, like the corner of a room, one for each plane of rotation.
Covering all the directions you could turn your head.
Exactly. Inside each canal, there's fluid. When you start to turn your head, the bony canal moves, but the fluid inside kind of lags behind for a split second because of inertia.
Like the water sloshing in a cup when you suddenly turn it.
Perfect analogy. And this lagging fluid pushes against a little gelatinous structure inside each canal, shaped like a little sail called a cupula.
Okay.
And embedded within this cupula are the stereocilia of the hair cells. So fluid pushes cupula.
Cupula bends the stereocilia.
You got it. And just like in hearing, that bending opens the ion channels, creates an electrical signal, and tells the brain via the vestibular nerve, hey, the head is rotating in this direction.
Same mechanism, different trigger. Amazing reuse of a design. What about the other parts, the otolith organs, utricle and saccule?
Right, those handle linear motion like accelerating forward in a car or going up in an elevator, and also sensing gravity, like just knowing which way is up when your head is tilted.
How do they manage that?
They have a really neat trick. On top of the hair cells in the utricle and saccule, there's a gelatinous layer. And resting on that layer are tiny, dense crystals made of calcium carbonate. They're called otoconia.
Botoconia, like tiny ear stones, seriously.
Yep, pretty much. Little weights. So when you tilt your head, gravity pulls these little otoconia downhill, Or if you accelerate forward, inertia makes them lag behind slightly.
And because they're sitting on that jelly layer,
Their movement, their weight shifting, drags that gelatinous layer across the stereocilia of the hair cells embedded underneath it.
which bends the stereocilia again.
Exactly. Bending opens channels, signal gets generated, sent to the brain via the vestibular nerve. This time telling the brain about linear acceleration or the static position of your head relative to gravity.
So canals for rotation using fluid lag, otolith organs for linear motion and gravity using tiny shifting stones.
Clever. Very clever. And your brain doesn't just use this vestibular info in isolation, right?
No, it has to combine it with other things, I assume.
Absolutely. It constantly integrates the signals from your vestibular system with what your eyes are seeing and also with information from proprioceptors.
Proprioceptors. Those are the sensors in your muscles and joints telling you where your limbs are.
That's them. Like knowing your arm is raised even with your eyes closed.
Hmm.
So the brain takes the vestibular data, head motion orientation, the visual data, what the world looks like, and the proprioceptive data, body position.
And puts it all together into one coherent sense of where you are and how you're moving in space.
Precisely. And that constant, updated picture is what allows you to stand, walk, run, catch a ball, adjust your posture. Basically, maintain balance and coordination without even consciously thinking about it.
Whereas running constantly in the background, powered by these hair cells responding to gravity and movement.
It's a nonstop process built on that fundamental conversion of physical force by the hair cells.
So pulling it all together, it seems the big takeaway here is this shared ingenious mechanism. Hearing and balance both rely on hair cells converting mechanical force vibrations, fluid sloshing, tiny stones shifting into electrical signals.
Yep, all through the bending of those stereocilia opening ion channels, they are the absolute crucial transducers, the go-betweens linking the physical world to our nervous system.
It really highlights the elegance and just how critical these microscopic structures are, allowing you to perceive sound and navigate the world with stability. Just amazing.
Truly architectural marvels at the micro scale.
So we've seen how these hair cells, often completely overlooked, are absolutely fundamental. Hearing, balance, two massive ways we interact with everything around us.
Couldn't do much without them working properly.
Okay, so maybe a final thought to leave everyone with. We've kind of mapped out these incredible detailed steps, right? Sound waves hitting the eardrum, leading eventually to tiny bristles bending, or a simple head tilt shifting microscopic stones, bending those same kinds of bristles.
Yeah, the whole chain reaction.
What does it really mean that our entire perception of sound, a whisper, a shout, complex music, or just our basic ability to stand up straight and walk around hinges entirely on the precise, delicate bending of these tiny, tiny structures hidden deep inside our skull? How much of our rich, stable experience of the world do we just completely take for granted, all powered by this incredibly intricate, almost hidden biological machinery? Makes you think, doesn't it?